Wherever it goes, it's going to reduce that molecule because it's a negative charge. But we don't want to confuse the fact that it's reducing the charge with the fact that it might reduce the energy. It doesn't. In fact, we have a higher energy level for a reduced molecule. A reduced molecule is going to have a higher energy level. Still talking about components of metabolism here. We're going to move to electron carriers. Electron carriers do just that. They serve to temporarily transfer the electrons from one molecule to another molecule. NAD is an electron carrier. FAD is an electron carrier. NADP is an electron carrier. Each of them has their own specific role in the metabolic process, but what we can say about them is that when they have an electron, they often will bring a proton with them. Now, there's hydrogen protons, hydrogen ions in the environment of the cell all over the place. They're just floating around there. But they have a tendency to want to hang on. See, if we added the electrons to this, we'd have a negative charge here, and the hydrogen proton wants to hang on to that negative charge. So it's going to go along for the ride. But what this tells us, when we have the hydrogen proton there, is that this molecule is now reduced. Okay? And we can say that this molecule has reducing power. So our electron carriers, when they have an electron, when they've picked up those electrons, they've been reduced, they have reducing power, meaning that now they're a high energy molecule, and when they pass those electrons on, they'll be oxidized, but they'll be passing on the energy from those electrons.
So whenever they have positive protons or electrons, they are high energy molecules?
Whenever, between these two here, when I'm talking about if I write it as NAD plus, this has been oxidized. So it's already given up its electrons, okay? When I write it as this with the H on the end, this one's picked up electrons, it's been reduced. So now it has reducing power, it's high energy. So just keep that straight between these molecules here. Whenever I write it with the H on there, that tells you that it's been reduced, okay? Because the proton is coming along for the ride. Yes?
So if it has an electron, then why is it plus, like the NAD is then, right? Why are they plus if they have that electron?
This does not have an electron though. It's got a positive charge, it needs an electron.
So when you say it's high, what did you call high energy?
This is the high energy molecule. This is not the high energy molecule.
Oh, okay.
Yeah. This one has been oxidized, and so it is ready to receive electrons. Okay, it can receive electrons, but when it receives those electrons, and here's the deal, you can see it's got a positive charge. What it's actually going to do is it's going to receive 2 electrons. So one electron cancels out the positive charge we see there, but the other electron then gives it a negative charge, and that's why the proton is attracted to it, and that's why the proton comes along for the ride. I won't expect you to tell me that, but that's what's going on, okay? So anyway, now we have a high energy molecule that can be reduced, can pass those high energy electrons on. One more component we'll talk about before we get into the pathways here. precursor metabolites. These are metabolic intermediates. These are molecules then that we'll find in various stages throughout the pathways, and they can, what that means then is that they can enter the pathways there or they can leave the pathways there depending on the need of the cell. So some precursor metabolites might need to be used as an energy source. So they'll enter into a metabolic pathway and be broken down. That same precursor metabolite might then also at some point need to be used in biosynthesis, and so it would leave the metabolic pathway and go into a biosynthetic pathway. So these precursor metabolites, they can be broken down further or they can be used to build larger molecules. They're what we call metabolic intermediates. Okay, so that's all of the components that we're going to talk about here. We'll find that metabolic pathways can be linear, so that you have a substrate, an enzyme that changes it to a product. That product then becomes a substrate that the enzyme changes to another product that becomes the substrate that the enzyme changes in a direct line. We can find that some pathways are branched. So you start out, then there's a molecule that splits in two, branching the pathway, and we'll find that there are some pathways that are circular, or cyclic, if you will. Let's start with glycolysis. Glycolysis is the most common pathway, initiates the breakdown of glucose. Your brain cells only operate on glucose. They don't work on anything else. You will break down muscle, convert it into glucose in order to keep the brain functioning. So, glucose, a 6-carbon molecule. In the process of glycolysis, and this is going to take place in the cytoplasm of prokaryotes and the cytoplasm of eukaryotic cells outside the mitochondria. We'll see the rest of these metabolic pathways for eukaryotes take place in the mitochondria, but glycolysis for eukaryotes takes place outside the mitochondria, in the cytoplasm. So we have a 6-carbon sugar, and what we do is we input some energy initially. We actually add some ATP in a couple of places. So there's two reactions here. In which we use some energy. We input some ATP, so the phosphate bond gets broken off, energy is released, and we move some electrons around. We do the same thing here. We move some electrons around and split the 6-carbon molecule into 2 3-carbon molecules. So it took some energy to do this. It took 2 ATP, in fact, to initially split the glucose molecule.
At the beginning, did you say the glucose, well, you're breaking down the muscle? I thought it was dangerous to break down the muscle.
Yeah, but it's also dangerous for your brain cells not to work. I mean, that's if you don't have any glucose going to your brain cells. This is off subject a little bit, but if you don't have any glucose going to your brain cells, your body's going to respond by breaking down your muscles to get glucose to those brain cells. That's all I was saying.
Because I was thinking it just break down the glucose, the sugar away from it. It's kind of confusing.
See me in lab. We'll talk about it in So we've input some energy. We've broken our glucose molecule into 2 3-carbon molecules, and now we can start to get some energy out of this 3-carbon molecule. Enzymes that are responsible for reactions, and there's a step of reactions to take place. Enzymes are going to move those electrons around. And as they do, they're going to generate a molecule of reducing power. So they're going to generate an electron carrier and
tell us what those are?
down here?
Yes, all of those.
Okay, so what we have going on here is we have an NAD molecule, one of our electron carriers, picking up electrons, now is reduced. So this is NADH. We also are generating some ATP. Remember, we put in some energy initially, now we're getting that energy back. In moving around electrons, breaking bonds, we're freeing up energy and capturing it in the form of ATP. So we do that twice in this series of reactions here that leads to pyruvate. So, at the end of glycolysis, we've taken a 6-carbon molecule and we've broken it down into 2 pyruvate molecules. These are 2 3-carbon molecules, pyruvate, and from that, we've generated 1 molecule, if you will, of reducing power, and 2 molecules of ATP for each pyruvate molecule that we generated. So this happens on both sides, right? So we really have 2 NADH, 2 ATP, 2 ATP, so we get 4 ATP, 2 NADH molecules from the exercise, and it only cost us 2 ATP. So our net gain, 2 ATP and 2 NADH molecules. Plus, pyruvate is a precursor metabolite. Questions?
So those 3 carbon so those NADH and ATPs are created by enzymes? That's what you just said?
Each step here is a reaction that an enzyme facilitates, okay? The enzyme is only facilitating the reaction, okay? In facilitating the reaction, you're releasing energy, okay? In this case, that energy is captured in the form of electrons with this electron carrier. In this case, that energy is captured in the formation of ATP. This ATP molecule and this ATP molecule are generated by substrate level phosphorylation. They've been generated directly from the breakdown of the glucose molecule. So this is substrate level phosphorylation going on right here.
Just in that little section under the 3-carbon substrate.
Right here. These 2 ATP have been generated by substrate level phosphorylation.
So the net gain is generated by substrate level phosphorylation.
These ATP are.
Oh, just ATP.
Yeah, those ATP are being generated by substrate level phosphorylation.
So glycolysis, and you have a total of 4 ATPs that are generated by the substrate?
You do, and it costs you 2. So your net gain is only 2.
2 are up in the cost, correct?
Yeah, it cost you 2 to get this started,
Right.
and you generated 4.
And the 4 that you generated after the substrate level phosphorylation
Yes.
are below after it splits into the 3-carbon pressure. Okay.
Right here. Okay, so now we have a 3-carbon molecule, pyruvate. If we needed it for biosynthesis, we could send it off in that direction and we could use it to build larger macromolecules. But if we don't, if we need it for energy production, then we're going to do that. And what happens here for energy production is that we take the molecule and we run it through what is called a transition step. So that it can enter into the next pathway of metabolism here. In this transition strip, this transition step, this 3-carbon molecule is going to be converted into a 2-carbon molecule, and coming off that conversion, we're going to produce some CO2 and some NADH. Okay, so it's got high reducing power. Here's our carbon that we lost. Now think, this happens twice, right? We have two pyruvate molecules that have been generated, so each of them will pass through this transition step. Well, this 2-carbon molecule can't enter into the next phase without a little help. There is a coenzyme, CoA, that will add to this 2-carbon molecule, which is acetyl. So what we end up with here is a molecule called acetyl-CoA. All the CoA is doing is help moving the molecule into our next pathway. And our next pathway is known as the tricarboxylic acid cycle, the TCA cycle, the Krebs cycle. Maybe you've heard that term before. It's a cyclic pathway. And so this 2-carbon molecule here